Welcome students. In this video, I will discuss about the Chargaff's rule, its exceptions and some numerical problems that appeared in various competitive examinations. The model structure of DNA, or commonly called the DNA double helix, was proposed by James Watson and Francis Crick in 1953, and they received the Nobel Prize in Physiology or Medicine in 1962 for their discovery. But probably you know that some previous findings of some other scientists helped them to solve the mystery of DNA double helix. One of them is Erwin Chargaff and his co-worker. Here I will discuss how Chargaff performed his experiment and how he analyzed his findings in a very easiest way. He treated the DNA sample with acids to remove the bases and then separated the bases by paper chromatography, and after the separation he measured the amount of each base with UV spectroscopy. In this experiment he isolated the DNA from ox. He isolated DNA from three different tissues like thymus, spleen and liver. He analyzed the molar ratio of all four bases. You can see that the molar ratio of all four bases in DNA are same no matter from where the DNA was isolated. So he concluded that DNA specimens isolated from different tissues of the same species have the same base composition. In this second experiment, he isolated the DNA from human and again from three different organs. But here, in one experiment, he used normal as well as cancerous liver tissue. But again, surprisingly, he observed the same thing. From this, he concluded that the base composition of DNA in a given species does not change with an organism's age, nutritional state, or changing environment. In his next experiment, he isolated DNA from two different species and determined the molar ratio of all four bases. Here he found that the molar ratio of adenine is not same in yeast and in Mycobacterium tuberculosis. And from this, he concluded that the base composition of DNA generally varies from one species to another. When the DNA of several species were analyzed, he found this. You can see that in all organisms, adenosine and thymine ratio is nearly 1, and on the other hand, guanosine and cytosine molar ratio is always close to 1. But the ratio of adenosine and guanosine and on the other hand, thymine and cytosine are not close to 1 in some organisms. From this relationship, he concluded that the sum of the purine residues equals to the sum of the pyrimidine residues. That means A plus G is always equals to T plus C. This quantitative relationship is called the Chargaff's rule. But all nucleic acid do not obey Chargaff's rule like single-stranded RNA and single-stranded DNA. Now I will solve some numerical problems from Chargaff's rule that appeared in various competitive examinations. In this problem, the nucleic acid was isolated from a microorganism and the base composition was determined. Adenosine is 35%, thymine is 15%, guanosine is 35% and cytosine is 15%. You have to say the physical nature of the nucleic acid. According to Chargaff's rule, we know that adenosine is always equals to thymine and guanosine is always equals to cytosine. But here, adenosine is not equals to thymine and guanosine is not equals to cytosine. So the nucleic acid may be single-stranded DNA or may be single-stranded RNA. So option C and D both are correct. Then in this problem, a double-stranded DNA was analyzed and you have to say that in which condition the molar ratio is always equals to 1. According to Chargaff's rule, adenosine is equals to thymine and guanosine is equals to cytosine. So A plus G is equal to T plus C. That means the option B is correct. And if we write cytosine instead of guanosine in this position and guanosine instead of cytosine in this relationship will not hamper. So option D is also correct. And in this way you can also solve this. The answer is B. And finally, in a double-stranded DNA, the percentage of adenosine is 15% and you have to calculate the percentage of all three bases. We know that adenosine is equals to thymine. So if adenosine is 15%, that means thymine is also 15%. So out of 100, 30% is adenosine plus thymine. So 70% will be guanosine plus cytosine. And again, guanosine is equal to cytosine. So 35% is guanosine and 35% is cytosine. So that's all for now. If you like this content, press the like button and subscribe my channel for more. And finally, thanks for watching.